we are going to set about our session 2 now, which, as I said earlier on, is focusing on IPM strategies. And we've done a huge amount of work on this in recent years, but we have also got a lot of recent work that's been done. So the program looks like this. We're kicking off with Michelle Fountain, who's going to be talking about Aureus as a predator for capsid bugs. Then Alicia from ADAS talking about push-pull strategies for midge control. Peter Seymour, similar push-pull approaches to control of thrips and strawberry. And then Celine Silva will talk to us about floral margins and their potential to support predators of thrips and other pests. And that will take us up to Ross George, who will talk about new approaches to aphid control. So I invite Michelle to come forward for our first presentation. Now, Michelle's going to be talking about Aureus. We know or have known for some time that Aureus is an excellent or can be a useful additional predator for western flower thrips to Neocelis cucumeris. And we wanted to find out whether Aureus could do a similar helpful job on capsid bugs. So, Michelle, welcome to the floor and take us through your findings.
Okay, thank you, Scott. Hopefully you can hear me and hopefully you can see the presentation.
Yep, that's absolutely fine. Thank you.
Yeah, is that on slideshow? Great. Okay, good. Thank you, everyone. And yeah, I just wanted to remind you before I get started on the Aureus that if you remember it, in the beginning of this project or some years ago, we have been working on a capsid repellent, which Adam Walker has presented on numerous occasions. That has been very successful at reducing Ligus damage to strawberry fruits, and we were seeing up to 80% reduction in damage to fruit in organic strawberry crops. We're now working with the industry to commercialise that through Russell IPM. So that is available, but one of the things that we all know and we have learned is that we can't just rely on one method for each individual pest, and it's good to have a toolbox of control methods to choose from depending on your situation and the pest pressure in the crop. So as Scott has mentioned, I'm going to talk about Aureus predation on Ligus. I do feel slightly fake here because I think I only did one of the assessments. The work was led by Bethan Shaw, and she was assisted by Ben Bruff and Adam Walker and also Francesca Elliott. And I'd also like to thank Francesca for sort of supplying or providing the first set of these slides for me from which this presentation is taken. So just as a reminder of the damage that capsids can do, so in the bottom right-hand picture, you can see this typical cap-facing damage. This is quite severe damage. And this is caused by the European tarnished plant bug, but also by common green capsid in strawberry, and you also get damage in raspberry fruits and some tree fruits. The second thing to say is that we've had reports in the last few years that where growers are releasing Aureus for western flower thrips control, they are feeling that they're getting less Ligus in the crop, so less capsids in the crop. We also had a bit of pilot data from the AHDB project SF147 that was also suggesting that where we were getting reasonable numbers of Aureus, we were getting fewer capsid bugs. So as I suggested, Aureus levigatus is used for controlling western flower thrips. It's normally released in the summer months when it's effective, and it feeds on the juvenile stages of thrips. So the question we wanted to ask is it also giving some additional benefit by predating juvenile stages of Ligus ruvula-pennis? So all of this work was done through laboratory testing. So we're just looking at looking for some initial kind of pilot data and feasibility studies. So all the Ligus were field collected and reared in a laboratory setting on a diet of French beans and pollen. And they were kept in these large cages. Here we've got some pictures of the eggs that the Ligus adults or Ligus females lay on the beans. So this is under a microscope, and you can see they look quite similar. The caps of the eggs look quite similar to anthochorid eggs, and of course they are from the same order of insects. In the second picture, this is a newly emerged, and I think quite cute, but then I'm a bit strange, first instar of Ligus rugilopennis, and we obtained the Aureus as the commercial product. So just going to whiz through the methodology. So we normally were introducing one Aureus into an arena, which was a petri dish with either the eggs or the nymphs of Ligus rugilopennis at different stages. The controls had Aureus the controls had capsid bugs, but with no Aureus, so that we could look at the natural mortality of the Ligus bugs. And we used either third instar Aureus or we used adults, but the data I'm going to present is just the raw data, unanalyzed and total numbers. So we've combined both the third instar Aureus and adults. And the other thing that I'm going to do is the assessments were done after 24 and 72 hours, but I'm just, for time's sake, just going to present the 72-hour assessments. One of the other things we looked at with the Ligus eggs is we looked at the behaviour of the Aureus around those eggs, and for this we used a computer system called EFOVision, which I will go on to explain later. And what I don't have time to present today is the predation on the actual eggs themselves, and this is actually being analysed by our biometrician at the moment. So I'm going to focus on the EFOVision for the egg predation and the predation on the different instars of Ligus rugilopennis. So you can see the picture on the right hand side, you've got the first through to the fifth instar, and in brackets these are the number of replicates, which is quite high, as you can see, that were run for each experiment. So moving on to how Aureus behaves around beans which have had Ligus eggs laid in them. So here you can see the EFOVision setup. So it's a camera looking down onto a petri dish, and in one side we have a bean with no Ligus eggs, and on the other side the bean with Ligus eggs. And this system actually tracks the activity of the Aureus bug, and it looks at duration around the bean, and it looks at how often the Aureus moves away from the bean, etc. So we've got lots of data to analyse here. But I'm just going to show you a very quick summary of the first three replicates, which are being run by Francesca. And the grey bar indicates the amount of time in seconds spent around the bean without Ligus eggs. And the red bar is the amount of time spent around the bean with Ligus eggs. So it looks like Aureus is orientated towards those beans with Ligus eggs in them. Now moving on to predation of the capsid nymphs, and just as a demonstration of the sort of the size, the ratio of the sizes between Aureus and Ligus, you can see this on the picture on the right hand side. There's a fifth instar Ligus rugilopennis in the blue circle, and you can see it's quite large in comparison to an Aureus nymph in the red circle. So the predation on first instar Ligus, you can see here on the left hand side, and all these charts will look pretty similar. The left hand side we had 100% survival where Aureus wasn't introduced, and again this is just total raw data, this hasn't been properly analysed. And then on the right hand side where Aureus was introduced, the Ligus mortality was just under 30% for first instar nymphs. And here again for second instar nymphs, we're looking at around just under 25% mortality where Aureus was introduced into the petri dishes. I've grouped the third and fifth instar together because the results are fairly similar, and you can see that the mortality of Ligus is not quite so high as the earlier stages, so around 4 or 5% mortality. And then the fourth instar, again, we got around, I guess, if we minus that off, it's about 15% mortality of fourth instar Ligus rubulapennis, where Aureus is introduced. So in conclusion, just as a warning, this data hasn't been analysed, and we think this is the first time that direct interaction between Ligus and Aureus has been investigated. All experiments were done in the lab, so these are ideal conditions where the Ligus nymphs aren't able to escape from the Aureus, so the Aureus has the best opportunity for being able to predate. But despite this, the mortality of the Ligus nymphs by Aureus was actually quite low. It was generally less than 25%, and it might be that they're able to predate on the younger instars. What's not clear from these studies is when the predation was happening. So it could have been happening as the Ligus were molting from, say, first to second instar or second to third instar. At this point, the insects are quite immobile, and that could be the point at where they're quite vulnerable. However, we were finding that the Aureus were orientating to beans where Ligus had laid eggs. So there seems to be some interaction going on between the two species. So although predation is low, there could be potentially disruption of Ligus in strawberry crops by Aureus, and there might be some semiochemical mechanism. And just to really add that the interaction of Aureus and Ligus does warrant further research, and we hope to be able to carry this on so that we might be able to exploit it in future IPM strategies. Finally, I would just like to thank the chairman of AHDB SF174, Stephen McGuffey, and in particular the industry representatives who guide us on what we decide to do in the project each year. And then also, of course, Scott Raffle and Rachel McGawley for all the support they've given us on various projects over the years. And that's it. Are there any questions?
Thank you very much, Michelle. And yeah, thank you for raising that issue. Project SF174 is an AHDB funded project, and although activities at AHDB are now winding down, this project will continue. I think Michelle wants it into next year, and potentially we could do some more work on Aureus if it's deemed appropriate.
Yeah, that's right. So we'll make those decisions with the committee in January.
Okay, thank you. One question here, could you clarify whether the Aureus were nymphs or adults? It seems more likely that adult Aureus would predate nymphs of Ligus. Can you comment on that?
So the data that I presented was actually a combination of adults and nymphs, and our biometrician is teasing apart the data at the moment. So yes, that's correct. It might be that the Aureus might be able to handle the Ligus better, but usually the nymphal stages of predators are more voracious because they're sort of going through a growing stage. But we hope to be able to tease this apart when the data is fully analysed.
Okay, thank you, Michelle. I think we're done there. So very grateful to you for presenting those results. And we will move on to our second topic of this session, staying with project SF174, which is a project looking at integrated pest management of softfruit pests. We move this time to cane fruit, and Alicia Bartel of ADAS has been working with others at Niab EMR on this project, and they've been looking at the push-pull strategies for midge control in cane fruit. Good morning, Alicia.
Good morning. Can you see me and my presentation?
We can indeed, thank you.
Excellent. So, hi everyone, I'm Alicia, and I'm going to summarise the research that we've been doing at ADAS, and also that Beth Shaw and her team at Niab EMR have been doing in investigating the efficacy of novel methods to control damage caused by raspberry cane midge and blackberry leaf midge, which are very damaging pests of raspberry. And we've been doing this using a pheromone-based push-pull strategy. So our objectives were to test whether numbers of raspberry cane midge larvae, which you can see in the top picture, and blackberry leaf midge damage, which is shown in the bottom picture, can be reduced by using Madripower sachets, which contain plant defense signal molecule methyl salicylate, to repel pests from the crop, and at the same time using pheromone lures to attract midges to sticky roller traps. We set up 2 trials, one in Norfolk and one in Kent, to compare 2 different populations. Here you can see the 2 different trial layouts at both sites. Each plot was 3 tunnels wide and 18 metres long in Kent and 20 metres long in Norfolk. The large plots accounted for wind direction, carrying the volatile compounds in any direction it could. We only sampled from an area that was 6 metres long from the centre of the central tunnels, however. The central tunnels were not sprayed with any insecticides by the growers, but the outer tunnels could be treated as standard. The fresh spawn was removed at the Kent site and left in place at the Norfolk site, specifically for this trial. Plots shown in blue were treated with push-pull, and those shown in green were untreated controls. I apologise that the colours look very similar here. There were 6 replicates arranged differently at the 2 sites due to the shape of the fields. This picture shows the setup of the treated plots in the central tunnels. Magipal sachets were hung 10 metres apart and 50 centimetres from the ground in the central row to repel the midges, and the sticky roller traps with lures embedded 4 metres apart were put in the leg rows of the central tunnel to attract the midges. And at both sites, the plants were grown in the soil. The Kent site was ahead of the Norfolk site as it's warmer down in Kent, so the first monitoring traps were deployed at the end of February, and the push-pull treatment was deployed at the end of March, after the tunnels had been covered with polythene and before the midges had started emerging. In Norfolk, the monitoring traps were deployed at the beginning of March, and the push-pull treatments were deployed in early April once the tunnels had been covered. This video shows my colleague Pete and I setting up the sticky roller traps in the tunnel leg rows of the central plots. In Kent, the first midges were caught on the 12th of April. You can see blackberry leaf midge here on the left, which is slightly smaller than raspberry cane midge, which you can see on the right. The midges are identified by their wing venation, and we taught the growers to identify these midges on sticky traps so that they could help us with the trap caps. Now, taking a look at the trap catches, this graph shows the population of blackberry leaf midge at the Kent site in the control plots only. You can see that there was a very obvious peak in numbers of 120 midges on the 7th of May. The raspberry cane midge numbers in the control plots are shown here and also peaked on the 7th of May, but you'll notice that the numbers are tenfold that of the blackberry leaf midge, which reached a maximum of 120 midges. Whereas here we can see around 1,200 midges were caught of raspberry cane midge. The blackberry leaf midge threshold was reached on the 30th of April in Kent, and 3 assessments followed this every 10 days. We counted the number of damaged shoots and damaged leaves, and you can see examples of these on the slide. And this is a good indicator of the number of larvae in the crop without having to take samples back to the lab. The raspberry cane midge threshold was reached on the 19th of April in Kent, and within 48 hours we made 10 centimetres splits in the canes with mounted needles to provide egg-laying sites for the females, as you can see in the video. And as the first flush of growth had been removed at the Kent site, splits were made in the old woody cane and in some lateral shoots on the first assessment, but more splits were made on each site visit as the new primer cane came up. Split canes were collected after 10 days and taken to the lab to count number of eggs, which you can see on the left, and numbers of larvae shown on the right under the microscope. And they can be found just under the epidermis of the cane. As you can gather from the pictures, the eggs and larvae start off very small, but as the larvae grow, they get larger and they turn red, and they can be seen with the naked eye on damaged canes. Now, to take a look at the statistically analysed results. These graphs show the trap catches for blackberry leaf midge on the top and raspberry cane midge on the bottom at the Kent site. Numbers in the control are shown in red, and blue represents the treated plots. There were significantly more midges caught in the control plots for both species between the 23rd of April and the 14th of May, which suggests that some disruption had taken place. And these graphs here show the damage caused by blackberry leaf midge to shoots on the left and to leaves on the right, split up between assessments 1, 2, and 3 going down the slide. The stars indicate that there were significantly higher numbers of leaves and shoots damaged in the control plots, which you can see in red on the first and second assessments compared with the treated plots in blue. However, when it comes to numbers of raspberry cane midge eggs, which are shown on the left, and results for the larvae shown on the right, there were no significant differences between the treatment and the control on any of the assessment dates at the Kent site. Looking at the results from the Norfolk site, this graph shows the numbers of blackberry leaf midges caught in the monitoring traps, and you'll notice that no more than 3 midges were caught on average from 6 traps, which is much fewer than the 120 caught in Kent. The first midges were found on the 7th of May, almost a month after they were found in Kent. And again, numbers in the control are shown in red and the treated numbers are shown in blue, but there was no significant difference between the control and the treated captures in Norfolk. In Norfolk, the raspberry cane midge threshold was reached on the 29th of April, which was 10 days after the threshold was reached in Kent. The canes were then split on the 4th of May and assessments followed every 10 days. There were significantly more midges caught in the control plot compared with the treated plot on the 24th of May, which you can see shown by the star on this graph. And a maximum average of 35 midges were caught, which is far lower than the catches in Kent of more than 1,000 midges. Here we can look at the damage caused by blackberry leaf midge to shoots on the left and to leaves on the right. Although it may appear that there was more damage in control plots, shown in red, compared with the treated plots in blue, on the second and third assessments as we go down the slide, when the damage levels were higher, but there was no statistically significant difference between the results. And the results for the cane split assessment in the lab showed great variability. Unfortunately, we didn't find any eggs or larvae on the first assessment and therefore made more cane splits when we returned to the site. We found low numbers of larvae on the second assessment from the original splits, with significantly more larvae found in the treated plots on the 24th of May compared with the control, as you can see in the bottom center graph with the star. We found no larvae or eggs again on the third assessment from the more recently split canes, so we did an extra 4th assessment on the 14th of June, looking at the original splits, which you can see the results of in the graph at the top in the center. But there was no significant difference between the treatments. There were very low numbers of eggs found on the 24th of May, shown in the graph on the right, but again, no significant difference between the treatments. So far, we can draw the following conclusions. At the Kent site, push-pull appears to be working to control damage caused by blackberry leaf midge, whereas numbers of raspberry cane midge may have been too high to be affected by mating disruption, at tenfold the number of blackberry leaf midges. In Norfolk, blackberry leaf midge numbers were very low, and no raspberry cane midge larvae were found on 2 out of the 4 assessments, so it's difficult to draw conclusions. It could be that raspberry cane midge numbers were too low in Norfolk and too high in Kent for push-pull to be effective, but more research is required to determine if this is correct. The monitoring traps results from both sites indicate that the midges were disrupted by the treated plots, as significantly more midges of both species were caught in Kent in the treated plots, and significantly more raspberry cane midge were caught in Norfolk on one day compared with the control plots. More information from this project will be available when the report is published. And I'd just quickly like to thank the AHDB Horticulture for funding this research, and thank Russell IPM and NRI for their in-kind contributions. And of course, the host growers who helped immensely with this project. It wouldn't have been possible without them. And my colleagues at ADAS and the team at Niab EMR. Thanks for listening. Are there any questions?
Thank you, Alicia. Very clear and quite promising results there. I suppose one question I have, or people might be asking the question, Magipal, am I right in thinking is that methyl salicylate?
Yes, yes, it is.
Yeah, the chemical in that. And how readily is that available to growers should they wish to use it?
As far as I know, it's already available from Russell IPM to purchase from today.
Okay, and similarly, the monitoring traps and lures, are they generally reasonably available to commercial growers?
I believe they are commercially available. I think the only issue is that you may have to pre-order them well in advance so that they can make enough of them. Okay,
that's useful to know. Yeah, just a comment from Bethan saying it seems like this approach is density dependent for the blackberry leaf image results. Can you comment on that?
That seems to be what it looks like from the results we had, and that was the one benefit of looking at carrying out this trial in 2 different sites with 2 different densities of populations, although we didn't know beforehand that they would be different densities. And I believe this work is going to continue next year to explore further as well. Okay, that's very helpful. We'll draw it to a close there as there are no more questions. Thank you, Alicia, for sharing that information with us. And yeah, as Alicia says, we will continue with this project SF174, and my colleague Rachel McGauley, who is with us today, will be overseeing that work as it continues next year. Thank you again, Alicia. Let's move on now to our next speaker, who is a colleague of Alicia's at ADAS, Peter Seymour. Again, I think he's known to many of you because he has spoken at this event in the past. Peter's done a lot of work on Western flower thrips for us and other thrip species in this project in the recent past. And he's going to give us an update on his push-pull approach to control of thrips in strawberry. Good morning, Peter.
Morning, Scott. Can you hear me okay?
We can, thank you.
Perfect. And can you see the laser pointer on the screen as well?
Yes, I can see that too. Thank you very much.
Brilliant. Okay, thank you. Okay, so hi everyone. For those that don't know me, my name's Pete Seymour. I'm a consultant with ADAS Horticulture and just a simple part of the wider team, including Jude Benison and a number of others that have been working on this project together. So I'm here to talk to you today about our sort of part within SF174, which is the IPM of soft fruit pests. Now, we've been looking at a push-pull system for the control of thrip species other than Western flower thrips that are damaging to strawberry crops. Now, obviously, the big question always is why? So some species of thrips are a difficult pest to consistently control. Western flower thrips, as you mostly will know, is readily controlled within IPM programs using the mite Neocoelus cucumis. However, there are other non-Western flower thrip species such as rose and rubus thrips, getting the wrong way around there, which the adults will often fly into the strawberry crops and are not controlled by Neocoelus cucumis. Now, some years we can even see this damage before other beneficials such as Aureus established fully, mainly due to the temperatures needed to get them going. And as a result, growers are often needing to spray for these thrips, but options are limited, especially with the amount of insecticides available. So our aim with this has been to develop a non-pesticide option for the control of these non-Western flower thrip species. Now, what we've come up with and what we've developed is this push-pull system. So it's designed so that it will pull thrips from the crop onto sticky roller traps and then pull and push them away from the actual crop itself. Now, we've got 2 treatments, an untreated and a push-pull that we've been testing. So it's purely one versus the other. Now, our plots are 20 meters long and span 3 tunnels. Okay, so they're really large. And this was to maximize the effects with the semiochemicals. Now, we've been using, similarly to the talk that Alicia just gave, we've been using Magipal, which is methyl salicylate, commercially available, and has been found to have repellent properties from Russell IPM. It also has the added benefit of being a generalized attractant for beneficials. So that's been one part of it. So that's our push. The pull is this OptiRoll trap, which you can see in the top right here. Now, that gives the visual lure, and then it's combined with a product called Lurum. Now, this is supplied by Coppert and is also commercially available. Now, that acts as a generalized attractant. It's been found to have attractant properties on a lot of other thrips species, including Western flower thrips, so it crosses a wide range, but we don't have any data presently on whether it has an impact on rose thrips, and that's one of the things we wanted to find out here. So you can see the general outline of the plots that we were using, and this is how it would sort of try and scale it up. Now you've got roller traps that are going into the leg rows and are designed to pull those thrips away. Now we had 2 sites with this trial work, and we designed it so that we would monitor both and set up the push-pull at both, and then whichever site was finding the greater number of thrips that we could then extend the monitoring for longer at, we would then select. We then we had already confirmed that rose and rubus thrips were present at these sites last year and at the start of this season as well, to make sure that we weren't just finding a site with Western flower thrips. Now, this is a very quick run through of what we were doing, but we were essentially looking at thrips presence and adults and larvae in the flowers. We were also looking at their presence on the traps. Now that was important so that we could tell if they were attaching themselves to the traps, but also very importantly to see if they were leaving the crop itself and if we were getting lower numbers there. Now we were also looking at percentage fruit bronzing. Again, we want to make sure that these sort of methods are actively reducing the damage seen in crops by growers, and that's the fundamental point. And we then were looking at the beneficials, so things like Aureus and Aeolothrips and others, especially with Magipal having a sort of attractant properties to some of the beneficials. Now, this is all very preliminary data. We've got a huge amount of data that we're working through at the moment. Both of our sites had active grower IPM in place. Now, our site 1 was very suitable, but unfortunately, unfortunately for us, we saw low numbers of thrips per flower at this site and low levels of bronzing. Good for the grower, obviously, but that meant that we ended up focusing a greater number of assessments as planned on the second site. Now, the second site, we carried out more assessments, and we also saw high numbers of thrips per flower, and we also saw high levels of bronzing seen in those plots. These photos on the right here aren't from previous years, they are actually from this year in that crop. Now, we do have a lot more data to crunch through, and we're barely scratching the surface of it, so there will be more details on this in future. Now, we also, on top of that larger scale push-pull trial, we also did a smaller trap trial, which is testing these push-pull components on their own. Now, this was a smaller sort of efficacy trial just held at one site, and that had 4 treatments. Now, this was the untreated to act as a comparison, Magipal again, which is the pest repellent and natural enemy attractant, and then we had the Lurum as a thrips lure. We also had another product called Thripnock, which is a new thrips lure that wasn't present last year when we started the push-pull, that Russell IPM are starting to sell. So we wanted to include that as well to get some early data on whether it affects these species. Now, as I mentioned, we set it up at one site. So this was at site 2, where we were seeing high levels of thrips and damage being seen. And we used high levels of replication because thrips numbers can be very, very variable. So we wanted to make sure that we could get the best data we could out of this. We set the traps up, and you can see in the photos here, it was a single sticky card trap mounted on a cane horizontally, and it had for the treatments which had a semiochemical, this was then mounted underneath the trap. We then put the traps out, monitored them after a week, and decided that we were going to leave them out for 2 weeks, and then came back and collected them. We've then recorded the numbers and species, the numbers of the thrips that were being seen on there, as well as any other beneficials that we were seeing. So this is some very preliminary results from this small trap trial. So on the axis here, we have the total number of all thrips that were being seen. So this is combined, whether they were Frankliniella species or other species such as rose and rubus thrips. We haven't identified them yet, which is why we've grouped them. And then on this axis, you can see our 4 treatments. So we had the untreated, the Magipal repellent, the Lurum lure, and the Thripnock lure. As you can see, it does appear that we have a treatment effect with Lurum, catching roughly 2 to 3 times the number of thrips that our untreated traps were catching. We don't know the species of them yet, as I've mentioned, but we have removed them already and are in the process of IDing them. It didn't appear that Thripnook seemed to increase the numbers very much, and that Magipal didn't actually reduce them as much as we had hoped. But we need to break this down into species more and fine-tune the data. And so with the Thripnook, Russell IPM have data to show that it's as effective as lure at attracting western flower thrips to sticky traps. Again, around 3 times as many on traps without lures. But there is no data yet on the other species, such as Fuscopenis, which is also why we're doing this. So hopefully we'll be able to shed some light on that once we've ID'd the ones that are coming off these traps to get an idea on what's going on. The same with Magipal. Russell IPM have done a lot of preliminary work, and they have indicated that Magipal, is very useful in a push-pull strategy, but it's likely that that was mainly on western flower thrips. So again, we need to get to the bottom of what the species were on our traps here to determine if it was a species effect and whether Magipal is more effective on other things. So we also do have data on beneficials, which is still being pulled together. So that is going to be in the final report once we've sort of pulled all that together and interpreted it fully. So there is much more yet to come. I'd like to just give a massive thank you to the HDB Horticulture for funding the work. Niab EMR, who are obviously leading the project and have been very active collaborators to work with. Coppert and Russell IPM, who have supplied a lot of the products that we've tested here. And also our 2 host grower sites, who have been absolutely fantastic and have been incredibly helpful in getting these trials sorted, especially in difficult times. So if there's any questions, I'm more than happy to answer them.
Thank you, Peter. I recognise the fact that results are preliminary at the moment, but some interesting findings from that. One question we've got about regulatory: what is the regulatory position with using pheromones and chiromones in a push-pull strategy?
I will be careful how I answer that, because I will just say I'm not basis trained. So if I'm incorrect here, but as far as I'm aware, you don't need to have, there is not CRD approval related in using semiochemicals in crops, but I may be incorrect. So checking with your advisor is the best approach there. Okay.
Thank you. Just a comment from Bethan here. Magipal is commercialized as an attractant to beneficials rather than a repellent, just to clarify that. Thank you, Bethan, for doing that. Peter, I think you're going to be around for a bit, so if there's any more chat in the chat room, perhaps you're on hand to answer any further questions that come in for that, and some useful information coming in.
And more than happy to answer emails if they get passed on in my direction as well, Scott.
Yes, please. Please do forward anything retrospectively to me, and I can pass it on to Peter. In the meantime, Peter, thank you very much indeed for your excellent, clear presentation, which takes us on to our next presentation. And again, within Project SF174 that we've been talking about this morning, Celine Silva and her colleagues at Niab EMR have been doing some work over a period of time to look at the use of floral margins to harbour predators of thrips and other pests. And of course, there's this perennial argument about this. Are we attracting more pests in as well as predators by using floral margins? This is an ongoing project. I think Celine has presented some results previously, but the latest position as we stand in late 2021 is now going to be presented to you. Good morning, Celine.
Good morning. I hope you can see my screen and presentation.
Yep, we can. Thank you. Yes.
Yeah, thank you. Yes, I have presented some of this data before, and I'll present some new data that hasn't been shown before. Like Scott said, I'm going to talk about floral margins and their potential to support predators from thrips and other pests. So this is part of Project SF174, and the objectives that we aimed for were to record establishment of floral margins, also to see the ability of floral margins to support natural enemies, and also look into pests that could be using those floral margins. we also wanted to see if or which impact it had on the crop. So for that, we had 4 sites, and we have 4 sites. 2 of them were raspberries and 2 of them were strawberries. 3 were commercial farms and were sown in the first year of this project. And 1 was at Niab EMR and was sown in 2019. So the commercial farms that were sown the first year were not ready for assessment. So we started with the Niab EMR site that some of you might be familiar with, the wet center. Setups are a little bit different between the commercial farms and the wet center. So in commercial farms, you usually would have a large floral margin next to a large area of crop. At the wet center, because we have a smaller area of crop, we decided to do something different. And we had 4 different floral margins, and we established repetitions of those plots around the wet center. So we ended up with 8 blocks of 4 different treatments around the wet center. So we started, like I said, we started for the first year looking at the wet center. The plots that we have established at the wet center are sainfoin, so a plot just with sainfoin, another one just with chicory, then another plot that has EM1 mix from Emersgate, and then the unsown plot that was used as a control. So for the unsown plot, we prepared it as we did for the other plots, as we would sow something, but then left it bare. So any seeds that were there or that fell onto that ground would germinate as they would in a non-managed ground area around the crop. And we looked at margin establishment, we looked at natural enemies and pollinators, and we also looked at potential soft fruit pests. So the first thing we found was that, unsurprisingly, plots like the unsown and the mix have a longer flowering period because they have multiple species in them. So you can see that sainfoin here in red has a high density of flower in May, but then it decreases quite quickly. So the control in the unsown, having a bigger flowering period also supports a more diverse beneficial insects. So we also looked at beneficials and potential pests. We will call them herbivores, as we don't know if they are actual pests. And here you can see that most of the activity and the higher numbers were in May and June. So here you want to focus on the green bits. They are the beneficial outcomes of our assessment. And when I say that the mix had more anthocorids, it's always in relation to the unsown plots. So it's comparing an intervention to not an unsown plot. So in May, we had more anthocorids for the EM1 mix. We had less capsids also in the EM1 mix and also in the chicory. For June, we had more parasitoids in the EM1 mix. And we had also more odmera beetles, which are the ones in the top corner of the screen. They spend a lot of time on flowers and could be a potential aid for pollination. So the main pests we recorded, or I want to say herbivores, were aphids and capsids. The aphids we looked at were not known strawberry pests, so they didn't match any of the pests known to affect soft fruit. The capsids, we did the same. We had to do a different assessment for capsids, so we collected them so we could actually identify which species were there. And we found common green capsid, European tarnished plant bug, and potato capsid. So here, low numbers of European tarnished bug and potato capsid, a little bit more of common green capsid, and those were mainly found in the unsown plots and in sainfoin. So if you look at the mix, there's less potential to have capsids in them. We also looked at thrips and which species of flowers thrips could be using in the floral margins. So here we found 3 different species of thrips. We found Frankenella occidentalis, which is the western flower thrip, the Thrips tobacci, the onion thrips, and the Frankenella intensa, which is a flower thrip. All the other thrips that are not known as a pest for soft fruit, we included in the others. So that is just there for you to have some more information, but they are not considered as pests for soft fruit. So in May, everything very low, although we recorded those species, they are very low numbers. Then in June, you will see that all the graphs are in the same scale, so you can see when they increase the numbers. Here, again, western flower thrips, very low numbers. We have a bit more of onion thrips, and we can see that they are mainly using yarrow and wild carrot. And we also recorded Frankenella intensa in white clover. In July, again, western flower thrips, low numbers. We're still recording a bit more of thrips, of onion thrips in wild carrot, and we no longer recorded any of the flower thrip. In August, availability of flowers decreased quite a lot, and the only stone plot that we had flowering was chicory, which had almost nothing. We had oakbeet and yarrow, and here we only recorded a few specimens of western flower thrips. So regarding vegetation establishment at the wet center, all plots established quite well. We had more than 90% of establishment for samphire and chicory. We had more than 70% of establishment for the mix. And in the control, as nothing was sown, we can see that the main species there were plantain, yarrow, oakbeet, and there was also a bit of bare ground and dry and dead vegetation covering those plots. So in 2021, the commercial farms were then ready to assess, and besides doing what we've been doing at the wet center and looking at the flower margins, we also wanted to look at the crop. And that's what we did this year. We gathered information about the edge and then looked into the crop 5 meters, 10 meters, and 50 meters to see what impact the floral margin was having. So this is very preliminary data, and it's just an overview for you to know what to expect. The data has to be analyzed and it has to be looked at per species. This is just a mean of all species found. So in June, not much happening. We tried to sample in May as we did the year before at the wet center, but flower margins were barely flowering. There was almost nothing there because of the cold weather. So yeah, June was very low too. Then July, a bit more beneficials in the floral margin. We couldn't see, at this point, we can't see any relationship with the crop. But like I said, we need to break this down. Some species may give a different outcome. The same happened in August and September. So there was not a clear relationship with the floral margin and the beneficials or herbivores in the crop. We also collected flower samples for thrips identification. So we're hoping to supplement the data that we collected last year. We collected different flower species, so we'll have new data on flower species. And we also like to see if the commercial farm matches the data that we collected at the wet center. We'll also have data about the establishment of each flower margin. So to give a bit more information on how successful we can be establishing them. So in conclusion, all the plots around the wet center established well. And like I said, plots with multiple plant species have a longer flowering period. there was a high abundance of beneficial insects in May and June of 2020, and the flower mix was where there was a better number of beneficials in June compared to the other floral plots. Most of the potential pests that we found could be aphids or capsids. Regarding the thrips that we found, so still low numbers, but the only ones that can be mentioned are yarrow, wild carrot, where we found the onion thrips, and then wild clover, where we found the flower thrips. We hope that the data that we collected this year will give us a bit more information and will also allow us to see which species are better suited for soft fruit crops. I would like to thank the HDB for funding this work, and all the growers for allowing us to access their farms, and also to helping in managing all the floral plots that we monitored. I hope you like the presentations, and thank you for listening.
Thank you, Celine. Very helpful. Question to you, Celine. A few questions here. Celine, are you planning to extend this project and the potential benefits, disadvantages of tree fruit?
We did that. We also looked at apple orchards in another ADDB project that finalized last year, and we found some good increase of natural enemies where we had floral alleyways implemented.
Sorry, Celine, we got a bit of sound problem there. I think everybody generally heard that. Celine did sound rather like a Dalek to me. So maybe a sound issue there. We'll try another question. A question from me really is capsids, the situation with capsids. Some growers might be concerned to see the increase in capsids, but you can just clarify there. Am I right in thinking that capsid numbers went up in the untreated area as well? It wasn't just the trial plots.
So what we recorded was that the unsown, so what would be usually there if you don't manage it, has more capsids than a flower margin. So by implementing a flower margin, you have potential to decrease those areas where they are not managed, where any species is just left to germinate.
Okay, sorry, your voice is still very distorted, but we'll try one more. Michelle's just added a comment here that just to remind everybody, the Bespoke project has been extended to 2023, and you can find more details there. That Michelle's put a good link in there. Yeah, I just wanted to raise an issue about aphids. You did say earlier on that there were less, or the aphid numbers that did increase were not typical aphid species. I mean, there may still be concern because aphid is such a massive problem for the industry at the moment. Because we've lost things like calypso and pyrethrins as crop protection products in recent times. So the last thing growers want to do is increase aphid numbers further. And that may be worth looking at and monitoring in future years to detect whether there are any other aphid pest species and not just friendly ones. Niab EMR did some work a few years ago on tree fruit, didn't they, which Shaba Naji did, or looking at decoys for ants, so using sugar to distract the ants from climbing up the trees in the spring. I don't know whether that's something that we could pick up for soft fruit as well. The big problem with sugar was that it dissolved rather rapidly when it rained. But it may be something else worth looking at. I don't know whether that's been given any thought. Michelle's just come in with another comment. Wildflowers plots are very important for parasitoids. They extend the lives of parasitoids and the numbers of parasitoids, so they may be useful for aphid control. So anyway, just a few thoughts there. Celine, as your sound quality has deteriorated, I think we'll leave it there. Fortunately, we got through all of your main presentation without any problems. So thank you for presenting to us, and we'll look forward to hearing for more results from that project later. Okay, we're moving on to the final talk now of the morning session. Ross George. Now, I need to tell you that Ross is a student at Harper Adams University. But more crucially, he is the first of our speakers today who is a CTP student. Now, what does CTP stand for? Well, that's Collaborative Training Partnership. This is a scheme that was set up, which is a few years ago, about 4 or 5 years ago now. It is part funded by the BBSRC, British Biological Science Research Council, which is government funded, and also part funded by industry. And there is a fruit CTP scheme, which was set up by Berry Gardens in collaboration with Niab EMR. You can find a lot more details about the CTP studentship scheme for fruit by searching for CTP fruit studentship, and you will find a dedicated website with lots of information. We are on to the 4th round of intakes, I think, or maybe even the 5th of students. Ross, I think, is on the 2nd or 3rd intake. He's based at Harper Adams University, and I'm sure you'll all be very pleased to hear that he's going to talk about aphids and aphid control, because it's a massive problem for the whole industry at the moment. So Ross, good afternoon, I should say now, and welcome, and we look forward to your finding about what you've been doing on this.
Yeah, excellent. I trust you can hear me okay.
We can indeed. Thanks, Ross.
Excellent. Yeah, so thanks for that introduction. I'll be talking today about a couple of one experiment that I've done in 2 parts, I suppose. So as a quick sort of introduction, I'm sure everybody here already knows all of this, but just for anybody that's not quite up to speed. Strawberry and
Ross, can I
yes.
interrupt? Just sorry, very briefly.
Yeah,
we can't actually see you. We can see the presentation. We can't see you. I don't know if you can turn your camera on so everybody can recognize you.
How's that?
That's not working, but don't worry. Oh yes, we've got you now. Perfect. You were back in business. All right. Sorry, okay. Ross, I'll leave you to get on.
That's fine. Yeah. So as everybody here probably already knows, strawberries are very important, an important crop for the UK industry, but there's a number of threats to strawberries in the UK. So my project is focusing on aphids, the three aphid species that you can see here in the photograph. There's the melon and cotton aphid, the strawberry aphid, no prizes for guessing what that one feeds on, and the potato aphid. So these are all pests of strawberry crops. There's 30 aphids that have been recorded feeding on strawberry. Lots and lots of different pests. There's pathogens that pose threats to the crop as well. But my project is focusing specifically on these 3 aphid species. So how do they cause damage to the crop? Well, some damage is caused just by direct feeding damage. There's a photo here. You can kind of see the stylet of the aphid sort of piercing into a leaf. This is the underside of a strawberry leaf. But a bigger issue than the direct feeding damage is virus transmission. So there's four viruses that I've included here, which are spread by 1 or 2 of the species that I'm focusing on. So obviously, it's an important crop. There's a lot of threats. We need to find ways to control those threats. Now, pesticides have their uses, of course. They have their place, but some of the issues that we're starting to see, I think Scott mentioned earlier, approvals for some products are being withdrawn, so some products are being taken off of the market, and this leads to more of a reliance on a smaller number of products, smaller number of active ingredients or active substances. And this in turn leads to resistance. Now, some species, particularly Aphis gossypii here, the melon and cotton aphid, resistance has been documented, quite widespread resistance has been documented. But new products are being brought to the market with sort of different modes of action to sort of the older style chemistry. I've seen it referred to. But this raises the question of how effective are they? Do they actually work? Do they do what they say they do? Well, that's where I come in. That's where my project comes in. There's a number of products that are being used in this project. So Batavia is quite widely used in strawberry crop protection. And this works by inhibiting lipid biosynthesis. Basically, I think I've misspelled biosynthesis there. Never mind. This is what's called a systemic insecticide. So as you spray it on the crop, the plant, it's taken into the plant and transported throughout the plant, and then pests that feed on the plant are affected by it, and it inhibits their ability to develop. But there's additional products here that are being used, and you can see here they work slightly differently, and I've put a little star saying that they're physically acting. So they all have slightly different modes of action, as you can see here. But essentially, the way that they work is you spray onto the aphids on the crop, and it kills them, basically. So in these experiments that I'm about to tell you about, I used 4 products. So Batavia, which was kind of a positive control, AHDB 9811 and 9810, and then Flipper. And these 3 last ones here, these are all physically acting. So like I say, the theory is that once you hit the aphids with them, that's what kills them. Whereas with Batavia, you don't have to hit the aphids directly. As long as you get the plant, that should be enough to control them. And then down at the bottom here, I've got a couple of negative controls. Water, we all know what water is, and untreated. So in the experiment, I'll talk a little bit more about that just now. So I reared some aphids on strawberry plants, so the 3 species that I mentioned previously, and then essentially took excised leaflets from the strawberry plant, put aphids on them, put them in a petri dish and sprayed them. 10 dishes for each of the products or the controls, and 10 aphids on each. So that was a lot of aphids. And then over the course of 5 days, I recorded mortality to see how effective these products are at actually killing the aphids that they claim to kill. As I mentioned in the previous slide, there was the untreated condition. That was just everything was set up the same, but I didn't spray them, just as a real baseline. So that way we can see, is there any impact just from spraying them with water? Do we get any effect from that? Is it the act of spraying, does that have an impact? Here are the results. I'm really proud of these graphs. I think they're quite nice. So you can see here on the potato aphid, you can see this really big gap. So we've got the controls down here. This is the water in the untreated condition, and same with the strawberry aphid and the melon and cotton aphid. And you can see this big gap. It's even more pronounced with the strawberry aphid. So these bars at the top are showing you the mortality percentage as a result of the different treatment conditions. And you can see the legend over on the right. And I think this is great. I think this big gap here is fantastic, because we're seeing that actually with the controls, if you leave them alone or you spray them with water, you don't really get much in the way of mortality. But when you start treating them with these different products, we're seeing that actually we're getting quite high levels of control. So up here with Batavia, we're seeing about 100% control for the strawberry aphid. That's this blue line here. And what's really interesting, I think, is that these are taken the daily levels of mortality. And what you can see with the potato aphid and the strawberry aphid in particular is you can see this blue line, which is Batavia, the light blue line. It's a little bit slow to get going, but eventually it gets right up to the top, and we see really good levels of control. Whereas these physicals, the physically acting products, as I said, when you hit the aphids, you get quite a sudden jump in mortality as soon as you hit them, and it kind of just levels off. It climbs a little bit, but it's potentially, I mean, it's the same as what we're seeing in the controls. A little bit of an increase in mortality, and that could just be due to age. Yeah, the main thing to take away from these physicals is that we get a sudden increase, and then it kind of levels off. The melon and cotton aphid, on the other hand, is a little bit messier. It's not quite as clear cut, and the Batavia, which was really high in the other two species, isn't quite as high. Now, there's not been any reports of melon and cotton aphid exhibiting resistance to Batavia. The Spirotetramat was the active that I'm aware of. I'm not entirely sure what has caused this. I think with the controls, we're seeing quite high levels of mortality. And I think that that's potentially to do with the fact that all of these aphids were put on leaves which were the same age. So all of the leaflets that they were put on were 2 weeks old. And for the strawberry aphid and the potato aphid, that's not that much of an issue. But for the melon and cotton aphid, I'm sure we've all seen it. They tend to cluster on these young leaves that are sort of around the crown of the plant, and I think potentially we've run into a bit of a hiccup here, where the material that they were given, the plant material that they were given, wasn't quite optimum for them. And then that raises the question of, well, how do we take this experiment and translate it into something that's a little bit more field realistic? And the answer is, of course, well, that's just my results. As I was saying, yeah, there's no silver bullet product that's really effective against all of the species, but all of the products, they are showing some usefulness. But as I say, it's a lab-based study. It's not that realistic. So the next step is, of course, to make it slightly more realistic. So this is a polytunnel that I've set up. 6 rows of, or 6 columns rather, of 18 Koya grow bags. In each of these grow bags, we put 8 strawberry plants, and each strawberry plant was then infested, or each bag rather was infested with one of the 3 aphid species, and then treated with one of the 6 treatment conditions that I mentioned at the beginning. So the Batavia, the 3 physically acting products, and then the 2 negative controls. And then sprayed, sort of following the rules on the label, using the maximum concentration and the maximum rate per hectare, and did the same with the water, sprayed that at the same rate. So we've got a nice clean comparison. I've not analyzed these results yet, but preliminary results look like they're echoing what we saw, or they look like they're supporting what we saw in the lab. So these physically acting products, they reduce the aphid numbers quite quickly, but because it's so important to actually hit the aphids, anything that you miss, any of the aphids that you miss, they'll start to reproduce, and then populations will start to increase again. So it's really important to make sure that you get really good coverage, you really nail those aphids. Again, with Batavia, we're seeing similar results to what we saw in the lab. So it's taking a little longer. It's taking a couple of days to really get going. But once it does, we're starting to see a really persistent level of control. It just keeps going. And then this highlights sort of my last point here, that the physically acting products, you really want to be reapplying them to make sure that you do. From the results that I've got, it looks like you need to be reapplying them to make sure that you keep on top of these aphid numbers. So just a quick overall results slide. As I've said before, these are kind of the take home points. If it's physically acting, you'll get a good, quick effect, but it won't last very long. If it's systemic, it will take a little bit longer to get going, but it will last a bit longer. And then you're going to have to reapply things, or it seems you're going to have to reapply things that act physically in order to maintain control. And then that raises questions about sort of financial considerations. If you're reapplying these products, labels recommend weekly applications, which is quite regular. Batavia, I think, is every, I think you can reapply every 2 weeks, but a maximum of 2 treatments per season, which is a lot less, a lot fewer applications. So with these physical products, you have to ask, well, we're going to get through more. We're going to have to be out in the field doing more of these applications. But so far, I think the results are positive. I think it's showing that these products do what they say they do, and as they're marketed as being sort of friendly to natural enemies, I think that again that highlights more work that needs to be done. One of my favorite things about this experiment is that it's done such a great job of highlighting all of the other experiments that I need to do. So looking at dose response and aphid behavior, maybe using a lower dose of these products, maybe we don't get such great control of the aphids, but maybe actually that helps with natural enemies. Maybe they can pick up some of the slack. It could be that these products aren't killing them, but it could be repelling them. So lots of work, additional work that I want to do, that I'm excited to do, and that I'm excited to report back. And then just a quick acknowledgements. Thank you to my supervisors. Thank you to my industry rep, Harriet Duncalf. Thank you to the AHDB and to the CTP, as Scott mentioned at the beginning.
Thank you very much, Ross.
Thank you.
That's splendid. Your talk is very pertinent because, as I mentioned earlier, aphids have become a massive problem for the industry. And I think your results concur with what most growers and agronomists are finding, that the Batavia does work well and it lasts for longer. There is one question about saying what is considered long lasting. Well, I think with Batavia, weeks rather than days, whereas these physical acting products are more like days. But I think it's a problem. Growers are getting very frustrated because they have found, like you've said, that they need to repeat these physical acting products over and over and over again, and that can take time and money. But I think your other work that at the start showing in the laboratory that if you hit these aphids, it is killing them. I think that's really helpful information, because what it tells the growers is that it's all about your application. If you don't hit it, you won't kill it. And I know growers are frustrated by that because they can't always kill every last aphid in the canopy. But I think it does just reinforce what we were thinking, that you really have to kill these aphids. What's the next stage then, Ross? This next year, what's your next approach?
Next approach, I think largely in the order that I've presented them on this slide. So I want to look at dose response and see if there is a way of if you use a lower application, can you still get away with can you still get high levels of control? And look, it's quite a vague bullet point here: aphid behavioral responses. So potentially these products don't necessarily, start that again. Potentially at a lower concentration, these products won't give such high rates of kill, but it could repel the aphids, it could deter them from settling, it could impact their reproduction, and then there's value in that as well. And as I say, linking that in with natural enemies to see if these two sort of control measures can complement each other. And then mimicking the same sort of thing with the natural enemies. So in the lab, get a nice clean, what happens if we spray them directly? Is it going to kill them? Is it not going to kill them? And then taking that out into the sort of a polytunnel setting and having a bit more of a realistic understanding of how these things work.
Okay. One final question. Was there any resistance to the products noted?
Not that I have noticed myself. As I say, the Batavia or the melon and cotton aphid exposed to Batavia didn't give such high, or didn't exhibit such high levels of mortality. There could be a resistance thing happening there. Like I say, it's not been reported, and it's not something that I've looked into in any depth myself.
Okay. All right, we must draw this to a conclusion. My final comment being that Janet Allen has also mentioned how shallot aphid is also still a major problem for the strawberry industry and can cause a major problem. Just finally to say that my colleague Adam Doxford at AHDB has been trying, as many agronomists will know, to try and secure EMU authorizations for flunicimid, which is sold as main man, and he's been having problems trying to get that through CRD. They're still working on that, and we really hope that we can have flunicimid for growers for next year. It is lunchtime. We have just gone over slightly time. Ross, thank you very much for your time. I should just also say that for all these CTP students, the purpose of this work that they're doing is to secure them a PhD at the end of it, but also it gives them experience of working in research within the horticulture industry, and we hope that some of them will continue to work in fruit beyond the end of their PhD studies. but in the meantime, it gives very helpful results to us in the work they're doing. Thank you, Ross, and thank you to all our speakers, both in both first and second sessions this morning.